Very first, we are going to look at two structures at once. First is the testis and the other is the seminiferous tubule. So, the outer fibrous connective tissue covering of the testes is called tunica albuginea. Tunica albuginea gives protrusions inside that form the fibrous septa of the testes. These fibrous septae, they divide testes into multiple lobules, and by multiple I mean around 200 to 300 lobules. Inside each of these lobule, there's 2 to 3 seminiferous tubule. The fibrous connective tissue covering, that is the tunica albuginea, and its protrusion inside, that is the fibrous septa, they are supplied by the branches of testicular artery that forms its own layer called tunica vasculosa or the vascular layer. So the red one is the tunica vasculosa, the gray one is the tunica albuginea, the gray one protruding inside are the fibrous septa dividing testes into many lobules, with each lobule having 2 to 3 seminiferous tubule. So let's look at a slide of testes. So outermost connective tissue covering is tunica albuginea, which protrudes inside forming a fibrous septa. Both of them are supplied by the branches of testicular artery forming tunica vasculosa. Now, the septa, it divides testes into many lobules. This is one of the lobule of the testes. Inside individual lobule, there's 2 to 3 seminiferous tubule. These are the transverse sections of those seminiferous tubule. In between these seminiferous tubules, you can see interstitium that is filled with two stuff. Number one, the blood vessel, and number two, these spatial red looking cells. These red looking cells called the Leydig cells. Alright, now what we are going to do is we are going to take this seminiferous tubule and zoom into it. So this is an individual seminiferous tubule. These are the other seminiferous tubules present at its periphery. The seminiferous tubule, it's lined by simple squamous epithelium. Inside seminiferous tubule, there is a spatial type of cell that you need to know about in detail, and that's the Sertoli cell. Let's look at the Sertoli cell. So Sertoli cell, as you can see, is a tall columnar cell with irregular outline giving out appendages. The Sertoli cell contains an ovoid euchromatic nuclei. These are the non-dividing cells in adults. So these appendages that have protruded out, they connect with one another with the help of a spatial junction called the tight junction, forming the blood-testis barrier. Due to blood-testis barrier, the toxin from the blood is unable to enter the testes, and the sperm from the testes is unable to enter the blood. Alright. Now, these Sertoli cell, they are also given two names. Number one is the sustentacular cell. Sustentacular meaning supporting, because you can see that they are supporting the germ cells, as well as nurse cell, and for nurse and for nourishment. They are providing nourishment to these germ cells. Another thing you might have noticed is that the appendages of the Sertoli cell has compartmentalized the process of spermatogenesis. You can see spermatogonia in the lowermost compartment, then primary spermatocyte in compartment right after that, then secondary spermatocyte in compartment after that, then spermatids, and then sperms. The head of sperms, they are lodged inside the Sertoli cell. Okay, let's look into the histology as well. So here's the Sertoli cell, which is tall columnar with irregular margin having appendages protruding outside forming the blood-testis barrier with euchromatic ovate nucleus. Now, due to compartmentalization by these cells, by Sertoli cell, you can see the germ cells in different layers. So these are your spermatogonia, these are your primary spermatocytes, these are your secondary spermatocyte, these are your spermatids, and these are your sperm cells with their head lodged inside Sertoli cell. Okay, now outside seminiferous tubule. In the interstitium, you have second special cell called Leydig cell, and these Leydig cell appear red in color because they are acidophilic and they secrete testosterone. This is the histological slide that could be provided to you in your anatomy practical. You can see that here's the septa that is present in testes. This whole thing is one lobule. Here's the cut part of the other lobule. The lobule, they contain many seminiferous tubule, and in between these seminiferous tubule, there are these red cells called the Leydig cells that produce testosterone. Inside seminiferous tubule, you can see the process of spermatogenesis going on. That is why you can see these round cells of different shapes and sizes arranged in layers. You have spermatogonia, primary spermatocyte, secondary spermatocyte, spermatids, and even sperm cells. Up next, we have epididymis. The tail of the epididymis is where the storage and maturation of sperms take place. Basically, sperm, they gain the ability of motility. However, sperm, they have to travel from the testes into the vas deferens, right? And that needs to be brought about by epididymis. Since the sperms, they have yet not gained the ability to move, epididymis, they transport sperm by the process of peristalsis. And to bring about peristalsis, epididymis is lined by a layer of smooth muscle. It is lined by a layer of smooth muscle. All right. See, since epididymis is highly convoluted in its body, if you were to take a transverse section, you can see multiple tubules of the same epididymal duct. If you look at a single tubule, you can see that from outside, it is lined by smooth muscle, which allow for peristalsis. Inside, there's the storage of sperm, and it is lined by pseudostratified ciliated columnar epithelium. That means if you were to look at the epithelium, it contains tall columnar cells with small basal cells, all arranged in a single layer. However, since nuclei, they are present in two different layers, they seem to be stratified. Now, the important thing is that these tall columnar cells, on their free surface, they contain microvilli, long, tall microvilli. And these microvilli, they are confused to be cilia. Since microvilli, they do not move, these cilia are confused to be stereocilia or non-motile. Stereo means non-motile cilia. So in reality, these are microvilli. However, under microscopy, they were confused to be stereocilia. So people have continued using the term stereocilia. This is the exact line from Witters. The principal cells of the epididymal epithelium bear tufts of very long microvilli, inappropriately called stereocilia. Okay, since there's microvilli, it is there in order to increase surface area. In surface area increment is required in order to increase absorption. What is getting absorbed here? Testicular fluid is getting absorbed here. Why? In order to increase the concentration of sperm. Why? In order to prevent sterility. How does it take place? So estrogen has estrogen receptors which gets activated by estrogen, and microvilli gets activated and the absorption of fluid takes place. Here's the histological slide. You can see epididymal tube right here with lots of sperm inside, and outside is a layer of smooth muscle for peristalsis, and the lining epithelium is pseudostratified ciliated columnar epithelium which contains stereocilia, which in fact are nothing but long microvilli. Vas deferens is a thick cord-like muscular tube. Since it is thick and it's a muscular tube, it contains multiple layers of muscle in its wall. See, usually the structures in the body contain 1 or 2 layers of muscles. Like epididymis, it contains 1 layer of smooth muscle. In majority of the portion of GIT, they contain 2 layers of smooth muscle. But here, there are 3 layers of smooth muscle. In GIT, you usually have outer longitudinal and inner circular. However, here there is outer longitudinal, inner longitudinal, and middle circular. So there's longitudinal outside, longitudinal inside, and a circular in the middle, sandwiched in between the two longitudinal layers. Let's look at the histology. So there's the lumen of vas deferens, here's the epithelial mucosa, there's lamina propria, there's your thick muscular layer, and outside there's tunica adventitia. The lumen is irregular or star-shaped. The epithelial mucosa is the same that is found in epididymis. Then the muscular layer, it contains outer longitudinal, inner longitudinal, and middle circular. Here's the slide. You have irregular lumen, which is surrounded by a thick layer of muscle. Seminal vesicle is really easy to identify. All you have to look for are these highly convoluted mucosal fold. So the mucosa is highly convoluted. You can see folds and folds of mucosa, and they are folded so much that they anastomose with one another. That is, they connect with one another and become continuous with one another, like branches of a tree. So highly convoluted mucosa. Inside seminal vesicle, there's seminal fluid, which forms 70% of semen. Obviously, this seminal fluid needs to be ejaculated out through the ejaculatory duct, and for the squeezing action to take place, muscle is required. So a layer of muscle, smooth muscle, is present surrounding the mucosa. Outside that is tunica adventitia. So all you have to look for is highly convoluted mucosa with a layer of smooth muscle outside. See, highly convoluted mucosa with a layer of smooth muscle outside. Prostate is a fibromuscular gland of the male reproductive system. It is made up of two parts, the stromal part and the glandular part. The stroma contains fiber and muscle, that's why it's called fibromuscular stroma. Due to the presence of muscle, the organ is formed when palpated. The gland is made up of two-layered glandular epithelium. So if we look at a histological slide, we can see glands, and in between these glands, there's your stroma. Since the stroma is made up of fibers plus muscle, it is fibromuscular stroma. As for glands, they are convoluted, and the lining epithelium or the glandular epithelium is two-layered. Let's look at the two layers. So there's a layer of luminal cells facing the lumen, and then there's a layer of basal cells. The basal cells that are in contact with the basement membrane are also called myoepithelial cells. They are spindle shaped or flat. They are called myoepithelial cells because they are like muscles, that is, they possess the ability to contract. On the other hand, the luminal cells facing the lumen of the acini, they are columnar with round nucleus, and the cytoplasm is stained pale. Okay, sometimes you can see an amyloid deposition in the acini, which is called corpora amylaceae, and the deposition takes place usually during pathology of prostate gland, like in case of benign prostatic hyperplasia. If you look at the histological slide, you can see that there's your dense stroma, which is made up of fibers and smooth muscles. It is red in color due to presence of lots and lots of smooth muscles. Then there are areas of glands with convolutions, and these glands, they contain two layers of glandular epithelium with deposition of corpora amylaceae inside.